Okay, this next series of videos is going to look at mutations and the effect that they have on the protein sequence. So what I have here is the same thing we looked at earlier, the same, if you looked at the transcription and translation video, this is the same sequence. But here, we're going to look at what happens if you change that sequence, if you have a mutation. So remember, we made an RNA copy by transcription, then we looked at the protein sequence by using our genetic code table. If a mutation happens, a mutation is a change in the DNA sequence. So in this case, the mutation we're looking at is here in the original sequence, we had a TA in the DNA, which was translated into a U, transcribed into a U in the RNA. Okay, but here there's been a mutation in the DNA. And so the DNA sequence, instead of having a T at this position, has an A. And that means the RNA, rather than a U, has an A in this position. So in order to figure out what kind of mutation we have to look at the protein sequence. We cannot tell. This just looks like nucleotides random to us. So we can't really tell what's happening until we look at the protein sequence. So what we're going to do is translate this protein as we did before. Again, always starting with the start codon, which is the AUG, which codes for methionine. And then we go to the second codon, which is CGA, still codes for arginine. And now we get to our mistake, right? This next codon used to be UCU, which coded for serine, but now it's ACU. So if you look at your genetic code table, you find out that ACU codes for threonine, not for serine. The next codon's unaffected, so again, it will code for serine. And now we hit our stop codon. So notice that a single amino acid has been changed. Instead of met-arg-cir-cir, we now have met-arg-3-cir. That's called a missense mutation. It means that one out of these 4 nucleotides has been mutated, changed, because the DNA sequence was mutated, the protein sequence is changed. And remember, because of proteins fold based on their side chains, we've changed now one of the side chains of the amino acids. We now have, instead of the serine side chain, we have a threonine side chain. And that could affect the way the protein folds. So the example of sickle cell anemia that you found in your notes showed you a single change, a missense mutation that causes a single change in one amino acid can cause a genetic disease because it can cause the protein to fold wrong and not work correctly. So even a single change, even just a missense mutation in the DNA that causes one amino acid to be put wrong in the protein can lead to a genetic disease.